Hi, I'm Jacob, and I'll be talking about the paper that looked at the impact of astaxanthin on antioxidant status and the reproductive outcomes in patients with polycystic ovary syndrome. So, a bit of background information. Polycystic ovary syndrome is an endocrine and metabolic disorder characterized by various things, such as anovulation, which is when the egg doesn't release from the ovary, infertility, obesity, polycystic ovaries, and other things. It's often associated with the oxidative defense system becoming impaired, and current treatment options have various limitations within them, containing many side effects with very little efficacy, and thus this condition needs a novel and effective treatment option that addresses these issues. So, because of the defective oxidative defense system, which is present within these patients, which is shown to have an elevated oxidative stress levels, the use of an antioxidant supplementation has sparked a particular interest, such as the instance of using astaxanthin, which is an incredibly strong antioxidant. So, looking at the mechanism of action associated with polycystic ovary syndrome, or PCOS, we see that under stress-free conditions, the NRF2 pathway is inactive, whereby NRF2 remains bound to the KEAP1 protein within the cytoplasm, which induces proteasomal degradation. However, when there is dysregulation of the redox homeostasis and elevated oxidative stress, NRF2 is released from that KEP1, so over here, allowing for NRF2 to enter the nucleus and bind to the antioxidant response elements, or R in this case, located on the DNA. This induces transcription of associated R genes, such as HO1, NQ1, and SOD, and these genes enhance the antioxidant defense mechanisms, offering protection against free radicals, and in the context of PCOS patients, they protect the granulosa cells and thereby hopefully facilitate improved reproduction outcomes for these patients. So meta-analyses comprising of over 68 studies, involve patients with PCOS showing a significantly high level of oxidative stress byproducts, with further findings also illustrating that the antioxidant effects of AST are exerted through the activation of this NRF2 pathway, with laboratory studies also showing that administration of AST to the granulosa cells, AST is the antioxidant, to these granulosa cells activated this path, in addition to suppressing the oxidative stress in animals with PCOS. There are several studies assessing AST and the mechanisms associated with this NRF2 pathway. There's only now need for clinical trials to actually determine the effects of the biomarkers that I mentioned before with patients that have polycystic ovary syndrome, and that's what this study basically aims to address. What the researchers attempted to do was to explore how AST could influence regulation of key genes within this NRF2 pathway within the granulosa cells. And furthermore, they wanted to evaluate the levels of oxidative stress and antioxidant markers within follicular fluid and in serum, in addition to examining association between these markers and the outcomes using assisted reproductive technology. So the study design was a double-blinded randomized placebo-controlled trial where eligible participants were allocated into one of two groups. Control group, obviously given the placebo, and an intervention group that received 8 milligrams per day of that antioxidant AST across 40 days. Serum and follicular fluid oxidative stress markers, gene expression, and assisted reproductive technology outcomes were measured, and the overall oxidative stress biomarkers were measured before and after the intervention, which assisted reproductive technology outcomes assessed post-retrieval. So when we look at this table, we measure the use of CAT, MDA, and SOD, and these are basically just all good measures of antioxidant defense mechanisms, with TAC being the total antioxidant capacity, reflecting the cumulative effect of these antioxidants present within the plasma and body fluids. Thus, we expect higher amounts of these to be associated with better patient clinical outcomes. The study found that CAT levels remained constant within the AST group, showing no significant difference before or after the intervention. However, upon assessing the placebo, there appears to be a significant reduction in that CAT concentration. This finding suggests that although AST might not raise the total CAT concentration as expected, it may instead prevent the reduction in CAT levels during the ovarian stimulation cycle. Then looking at MDA and SOD, there wasn't any apparent differences between the groups that were of significance. And with the TAC recording, there displayed a significant increase in the antioxidant levels, housing findings similar to that in previous studies. So now looking into the results of gene expression, we have figure 2, which depicts the mRNA expression of oxidative stress-related genes. We see that there was a significant increase in the NRF2 within the treatment group, in comparison to that of the placebo, which is here, which also showed that downstream targets, the HO1 and NQ1, were upregulated within this compared to the placebo, illustrating the antioxidant effectiveness in stimulating this pathway. So furthermore, they wanted to make certain that it was this particular pathway, the NRF2 path, being stimulated, and thus they wanted to assess the expression of the NRF2 protein in addition to the primary inhibitor, that KEAP1. They found that through the Western blotting, there was a significant increase in that NRF2 protein within the treated group, in contrast to the placebo. So you can see darker bands here compared to those very small light bands there. What this showed was that the AST was successful in stimulating this pathway, ultimately enhancing their antioxidant defenses within these cells. This being said, you'd typically expect that upon activation of this NRF2 protein, thus allowing NRF2 to be removed from that inhibitor, you'd have a reduced amount of the KEAP1, the inhibitor, due to its role in essentially impounding that NRF2 protein to prevent activation. However, in the study, it showed that KEP1 protein increased with the AST group, which was of quite surprise, as shown here. This suggests that this could be a feedback mechanism within this pathway, whereby evaluated NRF2 levels might have the cell increase the KEP1 expression to prevent the excessive activation of this antioxidant response, or perhaps that this pathway may be regulated more than just what's currently understood at the moment. So finally, upon assessing the outcomes of the assisted reproductive technology, the ART, we see that there was an increase in the rate of embryo and the rate of mature oocytes, as well as the rate of higher quality embryos, as you can see in this table here. However, we see that there was no detectable difference between the AST and placebo groups when looking at clinical and pregnancy outcomes. Thus, it depicts that the AST will be able to improve that oocyte and embryo quality within patients. However, it doesn't actually impact the pregnancy outcomes within these patients, which is quite important. So overall, what was depicted from this study was that the AST group was able to enhance their antioxidant defenses through this upregulation of the NRF2 pathway within the granulosa cells, with potential links being made between the antioxidant system and oocyte and embryo quality. However, due to there being no change in the pregnancy rates, which is what we want, there may be other factors that play a role in the outcomes of these PCOS patients. Significance of this paper demonstrates the potential role of antioxidants, such as AST, in enhancing particular stages of reproduction in these patients, but the lack of associated outcomes reflects the need for further understanding and research to help with success and fertility of women with this condition. So, looking at the strengths and weaknesses of this paper, they did a whole lot in terms of creating a very detailed experiment, measuring many outcomes, such as the oxidative stress markers TAC, CAT, MDA, and SOD, as well as gene expression through the studied pathway, the NRF2, HO1, and NQ1, as well as looking at the important measures of fertility, being of the assisted reproductive technology outcomes. The study showed major clinical relevance also, focusing on these PCOS patients and addressing the underlying issue in fertility directly. Furthermore, assessing the mechanism of action which may become compromised in these patients and tailoring the intervention and using antioxidant AST toward this was very well conducted. So the limitations of notice was obviously using participants from Tehran and Iran only, which obviously limits generalizability through genetics, lifestyle, and environmental settings. And approving upon this could have easily been done through recruitment of various locations, or if money was an issue, they could focus on including patients from various demographics, whether that includes those from just different races or those from different socioeconomic backgrounds, and recording them into the study. This would have ultimately enhanced the generalizability by a significant degree, and thus should have really been considered within the study. Also, it was a very brief limitation is that it was a relatively short study of 40 days with no follow-up on these participants. Although the dosage of 8 milligrams per day of AST is considered safe and apparently quite well tolerated, it should take precaution in the long-term side effects and interactions of other medications to assess its safety profile, particularly in those who continue to supplement this from the study. And finally, looking at confounding variables or factors, the study didn't consider important factors such as lifestyle or diet, where participants could be consuming vastly different quantities of these antioxidants, and thus impacting the major focus of the intervention in using the antioxidants to help combat defective antioxidant defense mechanisms. Some individuals who consume far more antioxidant-rich foods may have a naturally low oxidative stress levels, and thus reducing the overall impact on the antioxidant supplementation. Although reported by many studies that there can be a bias toward self-reported calorie intake, it only really needs to address antioxidant consumption to a certain degree to show significance. And thus simply having like a food questionnaire, ticking off foods that they ate that day may provide sufficient data to address this study's major limitation. So this study demonstrated that there was upregulation of this NRF2 pathway and thus an enhanced antioxidant defenses within these granulosa cells, showing that only oocyte and embryo quality were improved. With limited applicability toward the pregnancy rates in patients, it opens the gap that can be explored in assessing other factors which may contribute toward PCOS patients and fertility. Given the various limitations presented within the study, it would be good to assess these other factors which may contribute to these patient pregnancy outcomes. Thus, two major areas future studies could address is the assessment of the impact on individuals outside the current study's demographic, in addition to controlling confounding variables such as diets, and if they have any impact on the results whatsoever. Conducting these future studies will ensure that not only the NRF2 pathway is the one that becomes compromised and induces the PCOS complications within these patients, which allow for even further studies to address other mechanisms and interventional approaches toward patient outcomes. So sorry for the long presentation, but in conclusion, the antioxidant AST successfully targeted that compromised NRF2 pathway and improved the oocyte and embryo quality. However, given the vast amounts of limitations presented, more studies are needed to address if other confounding variables could have influenced the results or if other populations would obtain the same results, with even further studies needing to address if it's more than just this NRF2 pathway which is involved in PCOS patients. And that is the end of my presentation. So thank you.